Ribosomes are the factories of the cell. That's where the proteins are made. That's where the polypeptides are produced. And that's what we learned when we studied the parts of the cell. But now we're going to get into the parts of the ribosome. So ribosomes are made up of ribosomal RNA, and they're made up of proteins. They have two subunits and contain two binding sites for the tRNA molecules. So this is a cartoon representation of a ribosome. It's got a small subunit here and a large subunit. It's got two sites. It's got the P site where tRNA can bind, and it's got an A site where tRNA can bind. We can have two tRNAs bound at the same time. In between the two sites, in between the two subunits, we have a place for mRNA. So the mRNA will be right in between the two subunits. And this is what it looks like when it's in working. We've got this is mRNA going between the two. We've got two tRNAs that are matching codons. They match the codons to the RNA, and they have these amino acids. That's an amino acid, a string of amino acids, a growing polypeptide. So the square amino acid is the next one that's going to be added. So there is an initiation code at the beginning, and that codon marks the start of the mRNA message. So the translation of RNA into polypeptide can be divided into three phases. So the first one is initiation. Elongation just makes the strand longer, and termination ends the strand. Now, initiation itself occurs in two steps. In the first step, this is the first step, we get the mRNA binding to the small ribosomal subunit, and we get the first tRNA bound, and it's bound at the P site. The second step is to add this large ribosomal subunit. So let's look then at elongation. Elongation is just going to add amino acids to the polypeptide chain until we get to a stop codon. As the mRNA moves one codon at a time relative to the ribosome, tRNA with its complementary anticodon pairs with each codon and adds its amino acid to the growing polypeptide chain. Okay, so elongation will occur until we get to a stop codon. And that will terminate it. And now we're going to go to the PowerPoint. Should be ready. Okay. This is elongation. And we start with initiation has been completed. So we've got our mRNA is bound between the two ribosomal subunits. We've got a tRNA in the P site, and we have coming in codon recognition. This anticodon from the tRNA that's over here is going to go into the A site. We can get it to advance. There we go. The second step of elongation is going to be peptide bond formation. So this green arrow here, the circle amino acid is now going to be bound to the square amino acid. So we have a new peptide bond, and the peptide chain is now on, it's not on this codon, it's on the new one, the one that just came in. And our chain is one amino acid longer. Now we'll have translocation, and that tRNA will be moved from the A site to the P site, and we'll be ready then for another, we'll be ready for another tRNA to come in to the A site and make the chain even longer. So the flow of information here will have codon recognition, peptide bond formation, translocation, and then finally termination. The flow of the genetic information of a cell starts with DNA, goes to RNA, and finally to protein. The sequence of codons in the DNA via the sequence of codons in mRNA spells out a primary structure of a polypeptide. And the polypeptides will form the proteins that determine the appearance and function of the cell and organism. Mutations. Mutations can affect genes. Mutations are changes in the genetic information of a cell or a virus. It can be caused by errors in DNA replication, recombination, or physical chemical agents called mutagens. And we're going to look at the different types of possibilities. So the different errors that can occur. We can have a substitution error. We can have an insertion. So an extra base pair is added. We can also have a deleting of nucleotides. And that will alter the gene, and it'll have varying effects. So let's look at this first. This is going to be a substitution. Normal hemoglobin DNA has a CTT sequence at this particular location. If you have a mutation for sickle cell, instead of a T, you get this. You have an A at this particular location. That means that instead of, when you make your RNA for hemoglobin, instead of making a code that says GAA, which would give us glycine, you get GUA, and GUA spells valine, not glycine. And the result is that you have sickle cell hemoglobin, which was those funny looking red blood cells that don't go through blood vessels very easily and cause all sorts of effects. Okay, so let's look at the various changes. So this first one is a substitution. So instead of what should be here is a G, we get an A, and that causes us to get a serine instead of a glycine. We can have a nucleotide deleted. So if this is our DNA sequence for a normal gene, and one of our nucleotides are omitted, then you get a frame shift error. And instead of, we have lysine, and then instead of UUU, we get UUG. And all of the amino acids are changed. Similarly, an insertion error causes all the amino acids past the error to be altered.